electron transport chain. It is also called as the respiratory chain. The reason it is said as a respiratory chain because approximately 70% of the oxygen which is consumed by the cell is utilized by this electron transport chain for the production of adenosine triphosphate called as ATP. And it is a complex process, and the mechanism of this process is located at the inner mitochondrial membrane. And for the entire series of electron transport as well as synthesis of ATP, we have totally 5 enzyme complexes. So out of the 5 enzyme complexes, 4 enzyme complexes belong to electron transport chain, and complex 5 belongs to oxidative phosphorylation. So these enzyme complexes are the part of the inner mitochondrial membrane, which means these are embedded in the inner mitochondrial membrane, and their peptides of these complexes originate from 13 proteins which are encoded by the mitochondrial DNA, as well as from the nuclear encoded proteins which are imported into the mitochondria. So not only the synthesis of ATP is a primary role of the mitochondria, other than oxidative phosphorylation and transportation of electrons along the inner mitochondrial membrane, mitochondria also participate in diverse cellular processing. The main role is apoptosis mediated by cytochrome C and the production of reactive oxygen species, calcium homeostasis, as well as it also takes part in the process of immunity. So now let us concentrate on what are the components of the electron transport chain, which is also called as the respiratory chain. We have complexes as well as mobile electron carriers. These electron carriers may be the part of inner mitochondrial membrane, which means complexes, or may not be the part of the complexes. But overall, if we talk about these electron carriers, they are the flavin mononucleotide, as you can see in this table. The first one is the flavin mononucleotide, and next to the ubiquinone or coenzyme Q, and iron sulfur protein which is associated with FMN as well as cytochrome B, and the last family is the cytochromes. These are heme proteins, cytochrome B, cytochrome C1, cytochrome C, A as well as A3. All these are called as electron carriers in the electron transport chain. So these cytochromes are heme proteins. What's the meaning of this? Which means they contain heme iron. So the cytochrome A3 in combination is called as cytochrome oxidase, and besides heme iron they also contains copper. That's why these are called as copper containing heme proteins. Along the cytochromes which are mentioned in this table, only cytochrome C is water soluble, which means it is easily diffusible and plays an important role in the programmed cell death called as apoptosis. But whereas other cytochromes like cytochrome B, C1A as well as A3 are lipid soluble, that is the reason they are fixed to the components of the membrane, and that is the reason they are the part of these electron complexes. What is the structural organization of the components of ETC? So these components of respiratory chain actually do not function as a discrete carriers of reducing equivalents. So reducing equivalents means electrons, but they are organized into 4 different complexes what we are seeing here. So they are organized into 4 complexes, each of which act as a specific oxidoreductase. Along with this, we can see coenzyme Q as well as cytochrome C. Are they part of the inner mitochondrial membrane, or they are lying outside the inner mitochondrial membrane? They are not the part of the inner mitochondrial membrane. So the coenzyme Q as well as cytochrome C are not part of any complexes That's the reason they are not at all fixed in the inner mitochondrial membrane. So remember that coenzyme Q as well as cytochrome C are not part of any complexes and not part of the inner mitochondrial membrane. They just act like an electron acceptor or electron carrier. But other than the coenzyme Q as well as cytochrome C, all other components of the ETC are the part of inner mitochondrial membrane. So these complexes are arranged in an order of increasing redox potential. What is the meaning of increasing redox potential? Electrons always travel from reactants with negative redox potential to positive redox potential, which means we can clearly say that complex 1 has a negative redox potential, and compared to the complex 1, complex 4 has more positive redox potential. That is the reason electrons are traveling along complex 1, complex 3, complex 4 like this. So always you need to remember that redox potential is the main important factor which is responsible for the transportation of these electrons, because a reactant which has a negative redox potential is a best donor rather than acceptor of the electrons, and in the same way a reactant with a positive redox potential is the best acceptor when compared to that of the donor. That's the reason complex 3 is accepting electrons from coenzyme Q and coenzyme Q is accepting electrons from complex 1. Everything is according to the redox potential. That's the reason one important point you need to remember here, that these electrons always flow from negative redox potential to the positive redox potential. So that's why these reducing equivalents called as electrons flow in only one direction, that is from complex 1 to complex 4, because of the redox couple with the low redox potential is considered to be the better electron donor, where with the one with high redox potential is electron acceptor. This is the theory beyond the electron donor as well as electron acceptor. That's why these reducing equivalents called as electrons flow through the chain from the components of more negatively redox potential to the components of more positively redox potential. This is the basic understanding about the transportation of electrons along the complexes of the inner mitochondrial membrane. Let us talk about complexes in detail. The first one is called as complex 1. The complex 1 is called as NADH dehydrogenase oxidoreductase, or we can also call it as NADH dehydrogenase ubiquinone oxidoreductase. So the complex 1 is accepting its electrons from NADH, and this NADH which is present in the mitochondrial matrix may be produced from pyruvate dehydrogenase complex, may be a product of TCA cycle or beta oxidation, or maybe a product of shuttle mechanisms like malate-aspartate shuttle. Whatever may be the metabolic pathway which produces NADH, it has to enter into electron transport chain from complex 1. So the NADH so produced in the mitochondrial matrix by means of various metabolic reactions enters into electron transport chain at the complex 1. And here this complex 1 receives electrons from NADH, and in the complex 1 we have FMN as well as FES, which means the electrons are first taken up by FMN, later the electrons are transferred to FES, which is called as iron sulfur complex or iron sulfur protein, and from there the electrons are carried to coenzyme Q. That's the reason we are calling it as NADH coenzyme Q reductase. So the electrons are transferred first from NADH to FMN, from FMN to FES, that is iron sulfur protein, from FES to coenzyme Q, right? So once electrons are transferred to the coenzyme Q, at this particular step, 4 protons are pumped into the intermembrane space. Right, we can see very clearly here that 4 protons are pumped into the intermembrane space. So by this you should know that after the complex 1 is completed, where are the electrons now? Electrons are with coenzyme Q. What happened to the protons? 4 protons are pumped into the intermembrane space. So after the complex 1, once the electrons are with the coenzyme Q, how many protons are present in the intermembrane space? 4. Right? Now, let us talk about complex 3. Why are we skipping complex 2 here? Because whenever electron transport chain begins with NADH and H plus, the order is complex 1, complex 3 as well as complex 4. Complex 2 is not a part of electron transport chain if electron transport chain begins with NADH as well as H plus, because complex 2 belongs to FADH2, is the another product which is produced in various metabolic reactions in the mitochondrial matrix. So before discussing complex 3, let us talk about complex 2, then we move on to complex 3 now. So what is complex 2 here? The complex 2 is called as succinate dehydrogenase ubiquinone oxidoreductase, and the succinate dehydrogenase enzyme is a enzyme of the TCA cycle, and it is the only enzyme of the TCA cycle which is located in the inner mitochondrial membrane. All other enzymes of the TCA cycle are located in the mitochondrial matrix, because the only enzyme of the TCA cycle, which is succinate dehydrogenase, is taking part in the electron transport chain by means of complex 2. Right? So here both succinate as well as NADH are the products of the Krebs cycle, that is TCA cycle. So their electrons are transferred horizontally from complex 1 as well as complex 2 to complex 3 by means of coenzyme Q. Right? Let us see here. So FADH2, which is a part of the complex 2, or the complex 2 is a succinate dehydrogenase enzyme where FADH2 is produced. From there the electrons are carried to the coenzyme Q, from there the electrons are transferred to the complex 3. But when this process is happening, no hydrogens are pumped into the intermembrane space, but in the complex 1, 4 hydrogens ions are pumped, that is 4 protons are pumped into the intermembrane space, but this is not happening with the complex 2. Right? Now electrons are transferred from complex 2 to coenzyme Q, from coenzyme Q to complex 3. Here remember this, at the complex 2 no protons are pumped into the intermembrane space. Now whether it is from FADH pathway, whether it is from NADH pathway, now the electrons are located at the complex 3. Right? So what is the complex 3 here? Complex 3 is known as ubiquinone cytochrome C oxidoreductase. So this complex 3 contains cytochrome B and cytochrome C1. In between B and C1 we have another mobile carrier called as FES protein or FES complex. So from coenzyme Q the electrons are accepted first by cytochrome B, then electrons are transferred to the FES, then electrons are transferred to the cytochrome C1. This is the actual pathway of the electron transportation according to the redox potential. So how these electrons are transferred from coenzyme Q? From coenzyme Q, these electrons are transferred to the cytochrome B iron sulfur protein called as FES complex and to the cytochrome C1. From cytochrome C1, the electrons are transported to cytochrome C. Right? So during this process, once the electrons are transferred from complex 3 to cytochrome C, again 4 hydrogens are pumped, that is 4 protons are pumped into the intermembrane space. Now, let's review here. How many protons are pumped into intermembrane space if electron transport chain begins from NADH? From complex 1, 4 and from complex 3, 4. How many protons are there? 8 protons are there in the intermembrane space when the electrons are accepted by cytochrome C if electron transport chain begins with NADH and H+. But it is not true if electron transport chain begins with FADH2 because protons are not pumped into the intermembrane space by means of complex 2. So, by the end of complex 3, when the electrons are present or located with the cytochrome C, only 4 protons are pumped into the intermembrane space, right? Only 4 protons are pumped into the intermembrane space by the end of complex 3 if it is by means of FADH2. 8 protons are pumped into the intermembrane space when the electrons are present with cytochrome C if electron transport chain begins with NADH as well as H+. Now, so after the complex 3, let us talk about complex 4 here. So, complex 4 is the final complex of the electron transport chain, but it is the 4th complex of 5 complexes because complex 5 is called as oxidative phosphorylation. So, this complex 4 what we are seeing here is called as cytochrome C oxidase. Why are we calling it as oxidase? Because it is utilizing the molecular oxygen to accept electrons which are donated by the cytochrome C. Cytochrome C is donating electrons. To accept the electrons which are coming from cytochrome C, oxygen is readily available to catch those electrons by means of the complex 4 which is called as cytochrome C oxidase. And this cytochrome C oxidase which is known as complex 4 contains cytochrome A as well as A3. Now recently we will call it as heme A, A3 and copper A and copper B center because they are heme proteins, they also contain copper other than iron. So that is the reason we are calling it as heme A, A3 as well as copper A and copper B center, right? At the complex 4, oxygen is the one which is accepting electrons and it is the final electron acceptor of the electron transport chain. So remember that who is the final electron acceptor of the electron transport chain is the oxygen at the complex 4, that is cytochrome C oxidase. So the electrons are transferred initially from cytochrome C to cytochrome A, from there to cytochrome A3, from cytochrome A3 then it is transferred to the oxygen to form water molecule over here. During this reaction, complex 4, 2 protons are pumped into the intermembrane space. So if the electron transport chain is by means of NADH pathway, totally 8 plus 2 is 10. 10 protons are pumped into the intermembrane space. If the same electron transport chain runs with FADH2, that is complex 2, 3 as well as 4, we have only 4 plus 2 which is 6 protons are present in the intermembrane space, right? So because of 6 protons are present in the intermembrane space by means of FADH2 pathway and 10 protons are pumped into the intermembrane space by means of NADH pathway, we have a difference in the production of ATP because of the amount of hydrogen ions which are protons which are pumped into the mitochondrial matrix, because due to its concentration gradient by complex 5 is the one which generates 2.5 ATP molecule per NADH as well as 1.5 molecule per FADH2 molecule. So what is the complex 5 here? Complex 5 is the ATP synthase, which is the complex of oxidative phosphorylation. It is not a part of electron transport chain technically. And this is considered to be the smallest molecular motor which is present in the body. And using the energy released by the electron transfers, by complex 1, complex 3 and complex 4, because complex 2 is not pumping any protons into the intermembrane space, which creates an electrochemical gradient, right? This electrochemical proton gradient across the inner membrane. So because of this electrochemical proton gradient which is created because of this entire process of electron transfer, the ATP is generated by the complex 5 when hydrogen protons which are present in the intermembrane space flows back down their electrochemical gradient into the mitochondrial matrix, responsible for the generation of ATP from ADP plus inorganic phosphate at the beta subunit of the F1 component of the ATP synthase. Which means how many protons we have? If it is the NADH pathway, I will write it as 4, 4 and 2. For every protons which are flown during this ATP synthase process into the mitochondrial matrix, for every 4 hydrogen ions, for every 4 protons, 1 ATP molecule is produced. So when 4 protons are traveling down the concentration gradient, 1 ATP, another 4 1 ATP, another 2 half ATP. So if 10 protons flows down through the concentration gradient, right, how many ATPs are produced? If the pathway is from NADH, 1 plus 1 2 and half. That is the reason 2.5 ATPs are produced if it is because of NADH. In the same way, if you subtract the complex 1, because complex 1 belongs to NADH dehydrogenase, complex 2 is not pumping any protons into the intermembrane space. So if the electron transport chain is from FADH2, then how many protons are there in the intermembrane space? 6. For every 4 protons which are pumped into the mitochondrial matrix down its concentration gradient, again 1 ATP molecule and half ATP molecule, so only 4 plus 2 is equal to 1.5 ATP. That is the reason 1.5 ATP molecules is produced by means of 1 FADH entering into the electron transport chain. This is how the electrons are traveling along the inner mitochondrial membrane according to its reducing equivalence, according to the redox potential, finally to reach the complex 5 for the synthesis of ATP, and this is the electron transport chain.